The motor system can be thought of as a three-tiered hierarchy of motor control. There is the strategy stage, the tactics stage, and the execution stage. Execution, of course, just refers to the actual execution of a movement. But let's say, for example, picking up a cup of coffee, you execute that action. But how do you pick up that cup of coffee? What are the tactics you use? If you're right-handed, then you're going to likely use your right hand to pick up the coffee. You're going to use all 5 fingers to wrap around that coffee cup, and you're going to lift with a certain force. But what is the goal or the strategy? The strategy is taking a drink of coffee. And the motor system can be thought of as broken down into these 3 different stages. The strategy stage is supported by association areas of neocortex and the basal ganglia. The tactical stage is supported by primary motor cortex and cerebellum, while the execution stage is supported by the brainstem and the spinal cord. We won't really discuss this 3rd stage, execution, because much of the psychology has already taken place by the time we get to the execution stage. Therefore, we're more interested in the strategy and the tactics. Again, the association of areas of neocortex, which are roughly around here and back in the parietal regions. These are involved in the strategy stage. Also, the basal ganglia, which you can't see from this outside image, you have to slice into the brain to actually see it, is also involved in the strategy stage. The primary motor cortex, here in this teal color, is part of the tactical region, as is the cerebellum back here in the back of the brain. Let's start by considering 2 areas of the motor association cortex. These are the premotor area and the supplementary motor area. Both of these input into primary motor cortex, with primary motor cortex being at the tactical stage of processing. So to begin with, we'll discuss the premotor area, sometimes called the premotor cortex, which is involved in imitation and prediction. So let's start by considering an experiment illustrated here on the right. In this case, we have a monkey who has electrodes implanted into his premotor cortex. And when the monkey is sitting still, the neuron is firing at baseline levels. Each vertical line here represents an action potential on the part of this premotor cortex neuron. So again, when the monkey's sitting still, that neuron fires at baseline. What happens next is a light on this display is illuminated. And this is called the instruction stimulus. And it tells the monkey where he is to reach out to. He's been trained to reach to whatever light has illuminated. And when he does that, he gets a shot of water or orange juice or some rewarding stimulus. However, the monkey's been trained not to reach out immediately, but to wait for a trigger stimulus. That's when a second light appears and tells him that it's okay to move. So a delay period has been introduced into this experimental paradigm. And interestingly, this neuron in premotor cortex fires during that delay period, but not during the actual execution of the stimulus or of the action. Notice once the trigger stimulus comes on, the neuron eventually stops firing. If this were an execution neuron, then we would expect it to be firing while the movement is occurring, not to be firing while there is the delay period. The fact that this neuron is active during the delay period tells you that it must be involved either in the strategy stage or the tactical stage. We'll talk in a moment about how we know that it is at the strategy and not the tactical stage. As we said, these neurons are doing imitation and prediction. In fact, in this premotor cortex is where a type of neuron called mirror neurons were discovered. And these neurons are clearly involved in imitation. That is, mirror neurons are neurons that fire both when the animal performs an action and when he watches someone else perform that action. As such, they seem like good candidates for being involved in imitation. Indeed, we imitate each other all the time. There's something called the chameleon effect, where we tend to imitate our surroundings, namely people around us. If you're in a room of people, if one person scratches their head, then the other people are likely to soon thereafter scratch their head. If one person folds their arms, the other people are likely soon thereafter to fold their arms. We imitate each other all the time and we don't even know it. Experiments that demonstrate this will have a room full of people and they'll have a confederate in there planting certain movements like scratching the head, for instance. And afterward, you'll ask each participant, did you find yourself imitating the confederate? And people say, no, why would I imitate the person? But then they're shown video of themselves, and sure enough, they imitated the confederate. And it's rather surprising how much we imitate one another, and we don't even seem to recognize it. In terms of prediction, these neurons help predict the sensory consequences of an action. So that if, again, with the example of picking up a coffee cup, my premotor cortex neurons will predict if I execute this plan, then I should expect to feel a warm cup against my fingers and the weight of that cup. If that's what my premotor cortex predicts, then it goes ahead and executes that action. But if, on the other hand, my premotor cortex neuron predicts the sensory consequences will be that I'll hit the cup and it'll fall over and liquid will spill out, then I will cancel that motor plan and come up with a different one. So that's the prediction and the imitation aspects of premotor cortex function. As for supplementary motor area, it seems to be involved in behavioral sequences. One way we know this is through a TMS study by Gerloff and colleagues in 1997. They used transcranial magnetic stimulation to disrupt supplementary motor area activity during a sequence of piano key presses. So they would have participants play a melody. Let's just say, for the sake of argument, let's say it was Mary Had a Little Lamb, and they would play the sequence of piano key presses, and then as they were doing it, Gerloff would turn on the TMS to inhibit their supplementary motor area. And what he found was not that the people became paralyzed. That would only happen at the execution stage if you inhibited the brainstem and spinal cord. Instead, people were not paralyzed. They were still able to press the piano keys, and they could even press the correct piano keys. They just couldn't press it in the correct sequence. And interestingly enough, it took 1 second after stimulation for that disruption to occur, demonstrating that that supplementary motor area must be planning a movement about 1 second before you actually execute it. Now, how can we determine whether these neurons, particularly neurons in premotor cortex, are involved in strategy or tactics? Well, one study was done where monkeys were trained to use a pair of pliers to pick up a piece of food to eat it. And there were two different kinds of pliers. One pair of pliers was the typical pair where you squeeze the handles or you squeeze your hand in order to close the pincer to pick up the food. And we see the neuron fire vigorously when the monkey was planning that movement. But the monkey was also trained on using a reverse engineered pair of pliers where in order to close the pincers, you actually have to open the hand. So the two tactics between these are opposites. The first tactic involves squeezing the hand. The second tactic involves opening the hand. Yet this neuron did not change its firing rate. It fired the same either way. If this neuron were involved in tactics, then its function should change as a function of whether the hand was opening or squeezing. It did not change, given that the goal was the same, the neuron's firing was the same. This proves that this neuron was at the strategy level or the goal level of motor processing. Another set of brain regions that are important at the strategy level of the motor hierarchy is the basal ganglia, which is important for movement initiation. Basal ganglia include multiple structures that can be seen in the image here. We have the substantia nigra down here toward the base of the brain. We have the caudate nucleus up here, the putamen, the globus pallidus, or the two stripes inside of the putamen, and then the ventrolateral nucleus of the thalamus. This set of brain structures can be seen in their connectivity as follows. There's the substantia nigra, which possesses dopaminergic neurons, neurons that produce excitatory dopamine. This dopamine will excite the caudate nucleus and the putamen. When the caudate nucleus and putamen are excited, they release inhibitory neurotransmitter onto the globus pallidus. What the globus pallidus is normally trying to do is release inhibitory neurotransmitter onto the ventrolateral nucleus of the thalamus. And what the ventrolateral nucleus of the thalamus is trying to do typically is to excite primary motor cortex. Now, primary motor cortex is not part of the basal ganglia. It's the major output from the basal ganglia. And so if you're going to initiate movement, then you need to get excitation here at this final stage. That is, primary motor cortex needs to be excited so that it can then excite the brainstem and spinal cord to execute movement. And so the way to get a plus sign out here at the end is to start with a plus sign here. So as we say, the substantia nigra excites the caudate nucleus putamen. The caudate nucleus putamen inhibit the globus pallidus. Since the globus pallidus is inhibited, it cannot inhibit the ventrolateral nucleus. Since the ventrolateral nucleus is disinhibited, it can excite the primary motor cortex. This is the basic gist of how the basal ganglia functions. And one well-known disease state is associated with death of the substantia nigra, and that is Parkinson's disease. In Parkinson's disease, neurons in the substantia nigra are dying, and as such, they cannot generate the excitation to the caudate nucleus and putamen. Since the caudate nucleus and putamen are not excited, they cannot inhibit the globus pallidus. Since the globus pallidus is uninhibited, it can now inhibit the ventrolateral nucleus, which means the ventrolateral nucleus is unable to excite the primary motor cortex, and therefore movement is not initiated. And Parkinson's disease is marked by difficulty in movement, and in the advanced stages of Parkinson's disease, patients go into this kind of locked-in syndrome to where they really can't initiate any voluntary movements at all. So that does it for our discussion of the strategy stage of the motor hierarchy. Now let's talk about the tactical stage. And as we mentioned previously, the primary motor cortex and the cerebellum are involved in tactics. We're only going to bother to discuss the primary motor cortex for our purposes. Primary motor cortex is often abbreviated as M1. And it can be seen here in this red striped region. This sulcus here is the central sulcus, and right in front of the central sulcus is the precentral gyrus. And the precentral gyrus houses primary motor cortex. Wilder Penfield was the first person to discover the motor properties of the primary motor cortex when he went in during surgery and stimulated various parts of the primary motor cortex. And what he found out was that depending on where he stimulated, he could elicit movements in different parts of the body. This motor homunculus, as it came to be called, is a representation of the body in the motor cortex. Homunculus means little man, and it's as if there's a little man inside of your brain. Again, looking here at the central sulcus, we have the precentral gyrus, which is the primary motor cortex. If you take a slice of that and look at it from a coronal view, we can see the medial portion of the primary motor cortex, the dorsal portion, all the way out to the lateral portion. If you were to stimulate here, you would elicit movements of the toes. Stimulate here, you'd elicit movement of the hand. Stimulate here, you'd elicit movement of the mouth, etc. Now, one thing that's noteworthy here is that the face and the hand are particularly large in this motor homunculus. And that's because these two regions of the body are capable of very fine motor movements. If you consider the foot, the knee, the trunk, there's really not that many movements that they can make. Your knee, for example, basically, your knee can just move the bottom part of your leg forward and backward. And so, since it doesn't do a lot, it doesn't need many neurons to drive it. On the other hand, there are many degrees of freedom in the hand. Each of the fingers can be moved in multiple ways, and the hand can be opened and closed. Same with the face. There are many fine muscles in the face. It's been said that we can express over 1,000 different emotions with our face, and that indicates very fine motor movements. And for fine motor movements, you need lots of neurons to drive those motor movements. And that's why the hand and the face look so large in this motor homunculus. This is representing the fact that there are many neurons driving those particular regions.